So I would start to do it very fast because I think everybody's tired already, and this is the last talk of the day. So Laurent asked me also to do something about the, where is the laser here? Okay. About what has been done during these last 20 years in terms of understanding motions and interactions of molecules within cells. So before I start, I want to thank my sponsor, the lastminute.com, okay? And in particular, the CEO of the sponsor of the lastminute.com. So as they say, it's sans tracas, assistance par des experts, 25 years of holidays, you know. So before he came to me, I think he asked many different people, and they all say no, no. And there was only one left. Okay, so first, also I wanted to say this 20 years summarized in 2 days is, of course, there are disclosures, so there will be missing literature, of course. There will be imprecise statements, and of course, there will be whole sections of the field that are unshown and undiscussed. Okay, so now, if it works. Okay, too fast. Let me get back to the previous one, maybe. And there will be unexpected technical issue also. So here, as you all know, I guess, no, it doesn't work. Well, anyway, forget. So life is motion. So you're supposed to see movies moving here, but nothing is moving. But life is motion at different space and time scales. Okay, so it goes from a few nanometers and microseconds and even nanoseconds up to seconds and hours and years. And here up to micrometer and even above, of course. So if I go back 20 years ago, there was already a very nice review from Jennifer Lippincott-Swartz, Knapp, and Anke and Rossi about studying the protein dynamics in living cells. And within this review, what you could already see here, whoa, is that there was already the FRAP photo-bleaching technique, fluorescence recovery after photo-bleaching, that was mentioned. There were also all these variations of FRAP that occur at the beginning of the 21st century. So this flip, this flap, this photoactivation, and blah, blah, blah. And also they were talking about FRET, and they were also talking about the beginning of SCS, saying that it recently become accessible to cell biologists with commercially available microscopes. So all this comes from biophysics and physics lab most of the time. And if we go back in ages, what we could see is that these instrumental developments of FCS and FRAP were mainly released in the 1970s, and by the beginning of the 1970s for FCS and the end of the 1970s for FRAP. And all these were coming from the lab of William Watweb. And I mean, this guy really made a lot for the field in terms of molecule motion and understanding and analyzing molecule motion. And in addition to this FCS and FRAP technique, came also at this, let's say 10 years later, the single particle tracking, mainly coming from the lab of Michael Sheets, and as you can see from these 3 different papers. So as I said, there are 3 main methods to study the dynamic of molecules within cells: the FRAP, the FCS, and the single particle tracking. And to my knowledge, there's nothing brand new at the moment, okay? So if we compare, for example, here, as Marie showed you previously, here is the time of the first Mifobio and the time of the Imabio consortium starting. So this is the number of publications over the years, okay, in the FRAP field, in the FCL field, and in the single particle tracking field. And so here it's not in log scale, okay? So it's in normal scale, so it's nothing to do with GFP. So what you see here is that the maximum of the FRAP just happened, let's say, 5 or 6 years after the beginning of the Imabio consortium. For FCS, it's a little bit delay, and for the single particle tracking, it's still ongoing, okay? So I would say that FRAP is almost over nowadays. Well, FCS is still quite interesting, but certainly SPT is the future of the motion analysis in cells. Nevertheless, if we go back in time, we can see also that these two techniques, like correlation spectroscopy and single particle tracking, they were in fact already started in 1911, and the other one in 1914. And there's been a long time where people didn't talk about it before it came back to biology. So there are chances that these techniques are waves with times, and maybe FRAP will come again with new, I don't know, new something, I don't know exactly what. So this is also to show that at that time, there were a lot of theory ongoing in the field with all these famous physicists, and there were kind of back and forth between the theory and the experiments. So what FRAP and FCS and all these techniques gave to biology, in fact, they gave the possibility to switch from qualitative description of molecular motion to quantitative description of molecular motion. So this is a review, one amongst many reviews, talking about the FRAP here and the kinetic modeling. And in fact, it's always the same. So you do your measurement, and once you have done your measurement, you establish a kinetic model, and this kinetic model is based on fixed parameter. So the parameter is the geometry of what you're looking at, the geometry of what you've been photobleaching, and there are some variable parameters which are indeed what you're looking for and what you want to measure. So here, for example, binding kinetics, diffusion, and blah, blah, blah. So you make an analytical model, and you use this analytical model to fit the experimental curve that you have previously recorded within cells. And the idea is that depending on the parameter set or depending on the model, your fit will be better or not, and you will decide what is the best fit and what is the best model. So most of the time what is done, to be honest, is that you decide what is the kinetic model, so you know your fixed parameter, and you just look at the change in your variable parameters. When I say that, I mean that you have an a priori knowledge of what you're looking at. Either you decide this is diffusion, or diffusion plus binding, or diffusion plus advection, or things like that. So this is how it goes. And because it goes like this, what people immediately saw also during these last 20 years is that if you start to fit with something which is totally different, and it's called anomalous diffusion. So by anomalous diffusion, which is a full range of possible motions, it just says that the diffusion coefficient is different with space and time in some extent. So people immediately realize that you could fit any kind of curves with anomalous diffusion, and the best model will always be something which is anomalous diffusion. So you have a bunch of literature also released. So the very first literature, to my knowledge, in FRAP also comes from this Watt William Webb laboratory. And you have many people that have done that either for FRAP or for FCS, and of course also for single particle tracking. And it's a real ground play also for physicists here, as you can see. So you have the different random walk guides to anomalous diffusion, and looking if it's either ergodic or not, etc., etc. So again, what should be kept in mind is that the very first discovery of this is a French mathematician in the early 20th century that was named Levy. So nevertheless, what is very funny is that, of course, you will always fit better. But if you look carefully here, these are simulations of something which is not anomalous at all. This is only binding, unbinding plus diffusion. And depending on the binding, unbinding constant, sometimes you see that anomalous is better, sometimes you see that Brownian is better, sometimes you see that two population is better. So the conclusion of all this is that the time is not enough. So if you want to know exactly what's going on, you need to go at different space scale, and you need to look at what's going on at these different space scales. So quite rapidly, this is what people did. So they look at these multi-dimensional approaches to retrieve and or to map the diffusion coefficient as a function of space and time. So of course, with the technology, they start doing this above the diffraction limit. So rapidly, I name a few. So it comes from the Edidin lab, and it's in 1987, and also a French lab in Toulouse who did that a little bit later on. The spot variation FCS technique that comes from CIML and Fresnel lab was released in the beginning of the year 2000. And then also come the line or the circular scanning FCS Then the RICS, which is a two-dimensional fluctuation analysis. So in year 2006, then imaging FCS in the lab of Thorsten, who is here, and I think he will do something about this first in his talk and also as a hands-on. And on another topic came also the multiple particle tracking, which is the ability of track several and numerous particles at the same time. And I think that, as you can see here, all this is in the range of the beginning of the Mifobio consortium. And what is brand new now is that because of the single molecule imaging and the possibility to go below the diffraction limit, so all this has been expanded using either spot variation state FCS or scanning state FCS or SPT-PALM, or more recently the MinFlux and the RASMIN technologies. So with this, you can play by simulating numerically different possibilities in the space you want to explore. So here, for example, you compare what is free diffusion with a domain model where the molecule can enter, get in, and get out with a given probability, or a fence model. So the molecule is diffusing within the fence, and at one time it goes over and it hops over the fence. And if you do that, you can already predict what will be the behavior of this diffusion time that you deduct from the FCS, for example, curve. And if you look at the diffusion time at one single position as a function of the area you are covering, so you make measurements at different area. This is the spot variation FCS. So you can see that in the case of, for example, domain model, you expect to see that kind of behavior. In the case of fences model, you expect to see that kind of behavior. And of course, in the case of free diffusion, which is a total Brownian motion, then you will see something which is totally linear with the space you are exploring. So this is numerical simulation. And if you go now to cell and try to explore the different possibilities depending on the transmembrane protein. So for example, here you have the transferrin, which is supposed to bump into the actin fences. So you see exactly that you see what the simulation predict. You have this Taiwan protein, which is supposed to partition in so-called rough domains. So this is this model here. Sorry for the laser. This is this model here, and here clearly this is also what you see. So you have a positive intercept, and here you have something which is supposed not to interact with anything within the cell, and it looks like Brownian. Of course, all this is not that simple. I'll change this. And you see that even recently, Thorsten released a paper showing that to hop or not to hop, there are some exceptions in these so-called FCS diffusion laws. And we have also, in the lab, extended recently these FCS diffusion law in the case of binding, unbinding, and diffusion on the surface and diffusion on 3D. These are numerical simulation. This is the predicted results. This is the observation. And this is the comparison of this partition coefficient, for example, when we use these FCS diffusion laws and we use another assay, and we can see that we are quite close to what we can achieve with another kind of experiments. So now, what is the future and where we are now? In fact, now we are at the position where we can monitor simultaneously the spatiotemporal super resolution and multiparametric fluorescence microscopy. So it's a quite recent paper from the group of Thorsten, where you can have access directly to the mapping of the autocorrelation. You can look at the number and brightness to give you an idea of the average number of molecules within each pixel in the image. You can also do this SRF imaging at the same time, and you can also establish from these FCF diffusion laws that gives you information about the landscape you're moving in. There's been the same release also, well, let's say 10 years ago, approximately, in the group of Maxime Dahan by Jean-Baptiste Masson and Mohamed Elberi, where they used the original MTT algorithm to perform this inference map, so they can map the single molecule dynamics with Bayesian inference. And you can see here that they can map either the attraction potential, so the interaction, or they can map the diffusion, and they can have the entire diffusion map showing the different places. So here is the dynamic mapping of the cell. And of course, the ultimate now, which has been released a few months ago only, is that now we can observe directly motor protein stepping in living cells using the mean flux. So this has been released at the same time in Science. These are back-to-back papers in Science by the Jonas Rees group and Stefano group. And they clearly show that they can observe the stepping, the discontinuous stepping of the motor of the kinesin on a filament, and they can even observe that in 3D. So you have the dynamic mapping of the system within the cell. Okay, and in this field also, what is brand new, of course, like in every field here, this is the artificial intelligence revolution, which help us to go much faster in analyzing and even predicting also what will be the thing. Okay, perfect. Oh, so much? Okay. Okay, so now I switch to something which I'm not the best talking about that because I'm not an expert at all. Maybe Marc should have come or Emmanuel that I see could have done that instead of me. So the other big technique that has been used in the last 20 years in cell biology to decipher the molecular interaction is the use of FRET and film imaging. So again, if you go back 20 years ago, there was a nice paper from Tom Jovin in Nature Biotechnology about the FRET imaging. And in this paper, you can see all the possible methods that you can use to perform FRET imaging to have an idea of molecular interaction. So of course, amongst these old techniques, you have the one that are dedicated to fluorescence lifetime. So here you see the transfer by looking at the change in the lifetime of either the donor or the acceptor. But you have classical techniques which are just looking at the change of intensity of the donor and the acceptor. You can look at the change also of the anisotropy of this. So if you have one molecule which is giving energy before relaxing to another one, then you should see a change in its apparent anisotropy or polarization, depending on how you name this. And of course, there is also the donor depletion kinetics, which is you bleach, for example, the acceptor, and you look at the change in the donor, or you bleach the donor and you look at the change in the acceptor. Usually, you bleach the acceptor and you look at the change in the donor. So all these, of course, you all know, rely on the discovery of this Theodor Foster, but I think also a guy that is not often named, this is the Gregorio Weber, who did also a lot in terms of fluorescence polarization and energy transfer in a biological system. So this paper was released in the 1960s. So another big boss of the energy transfer is Enrico Gratton. So here on the left is one of the classical fluorescence lifetime imaging methods. So basically here, to illustrate here, you have one GFP which is far away from one M cherry. So the probability that there will be an energy transfer is quite low, because if they are not too much concentrated, of course, then they will not be in proximity and the energy transfer will never occur. So you have a mean lifetime, which is, so to be honest, I didn't check if they're looking at GFP of M cherry. I guess that they are looking at GFP, but, and if you know, just link these two different dyes by a linker, which you make shorter and shorter, you will see that the lifetime will decrease down to lower values where the FRET occurs. And of course, this is due to the fact that the energy is transferred from here to here. So at that time, you just made film imaging and you can reconstruct a map of the fluorescence lifetime and the variation of the fluorescence lifetime. But of course, in this case, you need to have something which is specifically dedicated, because if you want now to look at molecular interaction with two different molecules, for example, either you have a way to trigger the interaction and then you can follow what's going to change. But if you don't have a way to trigger the interaction, then you can imagine that anything might happen depending on what you're looking for. So this was the classical analysis. So this can be done by two different techniques, either by looking at the photon content, the time-correlated photon content approach, or just by looking at the phase shift in the light. I think what brings also new into the game from Enrico Gratton is to have a new analysis of this approach here and to use a phaser approach to fluorescence lifetime imaging analysis, where you can now have an idea, not only values of this decorrelation, desexcitation lifetime, but you can also have an idea of, you can plot these different desexcitation lifetime and look for changes during the interaction and stuff like this. And he recently published also a paper when he used this phaser FLIM combined to spectral fluorescence lifetime imaging that shows even increased possibilities. But I think the big switch recently is the possibility now to perform single molecule FLIM imaging. So you can simultaneously get information on the position and the fluorescent lifetime of one single fluorescence emitter. So this is mainly thanks to the apparition of this pad array. And as you can see here in the literature, so you have two different approaches, either in scanning approach in a confocal microscope or in wide field in classical microscopy. And if you look at that, then you see that the group of Jörg Anderlein and also here Ignacio that is present have shown the possibility to measure directly two different colors simultaneously lifetime within. So this is the single molecule lifetime, and of course, because this is single molecule, now you have distributions of lifetime depending on the environment of these single molecule. So because I am on single molecule, I think also one key point of the future is the single molecule FRET. And this is indeed an old technique. So the very first paper of the single molecule FRET was released in the 1996 in the group of Shimon Weiss. Here, after this was declined in two different technical approach, correct me if I'm wrong. One is the Alex SM FRET, so this is for alternate excitation single molecule FRET. So here you look at the fluctuation of the intensity by exciting either the donor or the acceptor one after each other. So when they come into the volume, then you can have fluctuation of the emission intensity of the donor and the acceptor, and you avoid the spectral bleeding through. And by analyzing this, you can deduce what was the amount of energy that was transferred from the donor to the acceptor. And there's another technique that has been developed, which is quite equivalent. This is the pulse interleave excitation in the group of Dan Lamb, that will give a talk in our module during this week. So this single molecule FRET is, to my believing, something very important for the next future. So there's still things to do, and there's a very interesting review paper that was published a few years ago about first-base dynamic in structural biology, all the challenges, perspective, and the appeal for open science practices. So share the data. And very recently, it's been shown that this single molecule FRET imaging could be performed in living cells. So here, as you can see, this is opening the single molecule FRET on its way to structural biology in live cells. So in fact, it opened new avenues on the possibility to look at dimerization of receptors and many other molecular interaction in real time in living cells. And if I remember well, I think it's this year or maybe last year that this paper was released. So where we know, so we are at the following point now. We can look at single molecule motion and interaction in real time in complex media. So we are close to reaching the nanometer and the microsecond scale, which is somewhere around here, you know. So we are close to something that is the molecular modeling of what we are looking at. So to my believing, the future will be to bridge the gap between the molecular modeling and the quantitative information from single molecule measurements in complex environments. So as you can see here, thanks to the coarse-grained molecular modeling, now these molecular motions that are modeled using pure physics behind it. So you start from the structure of the molecule, which is established by NMR or X-ray diffraction. And based on this, you can analyze all these forces of interaction of each atom. What you do is that you make an average force here. I simplify, okay? You make an average force here, and you consider that this average force here to the next atom here is modeled like this. So you have a kind of a force field, of a new force field that is able to establish, and then you let the system evolve with time, and you look at how it moves inside the system. And with this, you can achieve up to, well, millimeter is quite optimistic, but you can achieve up to micrometer and microsecond molecular motion in complex media. So just to finish, and it's okay. So to finish and to talk a little bit about what we're doing in the lab. So for example, here is a coarse-grained molecular modeling of a protein of the HIV virus, which is binding the surface of a lipid membrane. And if you look at this molecular motion, so the total simulation is 1 microsecond here, and from here to here, it's something like 20 nanometer. So if you look at this simulation, so you can see all these different beads that are representing the coarse grain of the structure of this protein that comes from the NMR, okay? So this is the real structure of the protein. So here you have a mirror state which is embedded into the system, into the lipid bilayer. And if you look carefully here, maybe I can go back there, can I? Not sure. Yes. If you look carefully here are two lipids that are trapped by the protein and that stay for a given time within the protein and exchanging, which show you that on average, if you look at the probability of presence of these lipids around the protein, and so meaning that you project the whole time of this and you look at where were the lipid, due to the rotation of the protein, you see that there is locally an enrichment in this lipid, which is the PIP2. Okay? So this is a model coming from the NMR and coming also from what is known from the lipid. Then you can perform the thing in a model system. So then you take this lipid, you make this is a real experiment, you make this supposed lipid bilayer and you inject the same protein and you look at what's going on by a simple method, which is self-quenching. So you start to see disappearing of fluorescence. And of course, what you can do is that you can label the different lipid that you put into the simulation here. And if you, for example, label the PIP2, what you see is that the PIP2 fluorescence disappear when you inject the protein. And disappearance is due to the self-quenching, meaning that these PIP2 are close to each other in a distance which is less than 10 nanometers. So in a simple model system, what we observe experimentally is what is predicted by the numerical simulation. And if we go no further with a more complex system, so we perform scanning state FCS here. We go in a T cell line. We either transfect or infect the cell with HIV. This HIV contains the same protein that is labeled with a GFP, and it helps us to find position where the virus is ongoing assembly. And now what we do is that we scan over these different positions. So now we have the position of the ongoing assembly of the virus. And here we look at the motion of the lipids, in red here, at the different position, either in proximity of the virus or far away from the virus. And if we do that, then we see that we have a very different diffusion coefficient of the lipid depending on the fact that it is inside or close to the viral assembly. Minute's over. Okay. And when it's far away from the assembly, then we have something which is much faster. And with this, we can look at a kind of confinement index that tells us what is the trapping of the lipid. And here, again, with the real virus in cell, we see that the PIP2 is trapped by the virus as it was predicted by the by the coarse-grained molecular dynamics. So, do I have 5 minutes left or no? Okay, so now I go fast. I could have shown you another example if it works rapidly, but it's too. So I will finish with this. Okay. So what has remarkably changed during these last 20 years in the field? Honestly, I think this is the screen of the computers. There's a clear difference in the screen of the computers. I mean, nobody can say the difference. So what remained the same during these last 20 years? The moon over the sea. I think even 20 years after, when you go just look at the sea and the full moon on top of it, it's still the same. So the question is that, what will be different in 20 years? So to be honest, I have no idea at all. I don't know what will be the field in 20 years. This I don't know. But I can scoop the organizer of Mifobio, because what will be very different is the moon, because the next Mifobio will be Mer de la Tranquillité, of course. This is the 5 to 14 January 2044, okay? Because it was booked before, so there was. And I think this is to my opinion, what is the future of Mifobio. Thank you.